Okay, well, welcome everybody to the Scientific Roundtable. Before we get started with today's speaker, I just want to remind everybody that the next Roundtable Scientific Talk is going to be on May 4th, 4 o'clock Eastern Time. And Alay Peligan's going to be from the Medical University of South Carolina is going to be speaking. Today, we're very fortunate to have Chou Huang. He's a professor of internal medicine, the Division of Nephrology at University of Iowa. Chou received his PhD from UCSF, and he did his nephrology fellowship at UCSF, and then did a postdoctoral training at Howard Hughes Medical University at UCSF as well, before he went to University of Iowa. And here I'm just talking. Most of his research has been in other areas of nephrology, and just within the past 4 years, he became interested in some of the work in polycystic kidney disease, particularly the study of the polycystin protein complex and the interactions between the ER and the mitochondria. So his talk today is going to be ER mitochondria disconnection promotes metabolic programming and cystogenesis in PKD. So Juan, please go ahead. I think you're on mute. How long?
Forgot about, okay. So we know primary ciliary dysfunction is important for in the pathogenesis of ADPKD, but we know the protein is expressed widely in the cell, particularly in PC2 in the ER is very abundant. Part of a function of ER resident PC2, of course, is facilitate ER ciliary polycystin 1 and 2 trafficking. But the stoichiometry is such that polycystin 2 is much greater than 3-to-1 ratio. So we've been interested to understand what are the ER resident PC2. We know early report PC2 regular ER calcium release, but more recently, we know polycystin 2 actually conduct more potassium than calcium, and this is more than one study shown that. So the question is, how does the K-permeable polycystin 2 regular ER calcium release? So we know for calcium to release from the ER, you need a parallel charge, or for example, by parallel anion CFTR. Alternatively, you can have charge exchange mediated by such as TRIG-B, which is a ER resident potassium channel that to exchange potassium for calcium. And TRIG-B has been well known. It exists in many cells to allow calcium release. It's been known if you delete TRIG-B, you impair ER calcium release. So what we think that polycystin 2 as the calcium potassium permeable channel maybe complement TRIG-B. So the question is why you need 2? As it turns out, we know the cytosolic calcium increase activated polycystin 2, but the TRIG-B is known that the activation require drop in the ER luminal calcium. So you can imagine that can be complementary. So this is the data we published by JIT a couple of years now. So if you look at taken from the polycystin 2 deleted cell from the consortium, and you look at the ER or the intracellular calcium release under calcium-free extracellular calcium-free solution, purinergic receptor activation release calcium in control, and then in the polycystin 2 non-cell is blunted. You can reverse that by express transgenic TRIG-B. Conversely, TRIG-B cell has impaired intracellular calcium release. You can reverse by expressing recombinant polycystin 2. So this tells you the two channels, as I say, function complementary to help each other. Even though they have a similar function but not identical. They are complementary. So if we take the PKD2 conditional knockout compared to control, you see cis. And if we express TRIG-V transgene, you can kind of reverse it. This is express conditional transgene at the same time of a PKD2 conditional knockout. So this is the cis index, so BUN are not shown here. So the question is, what happened, after ER calcium defect? We believe this is by affecting mitochondria. So I just want to tell you the TRIG-B, given its ER restrictive channel and can complement polycystin defect in the ER. So we use the TRIG-B transgene as a tool for ER resident PC2 function. So we know this is a nice work published by Boretta more than a decade ago in polycystin deficient or the PKD mutant cell. They have mitochondrial dysfunction with mitochondrial respiration defect. And at the same time, they undergo metabolic reprogramming, Weber effect, into aerobic glycolysis. Part of this is to go through pentose phosphate pathway to generate biomass for cell to proliferate. But with the mitochondrial dysfunction, mitochondrial membrane potential is going to collapse, is going to die. So the mutant cell actually evolve a mechanism called glutamine anaphylaxis, which they use asparagine synthase to convert glutamine into glutamate to fill back into the mitochondria. At the same time, to keep the mitochondrial membrane potential from collapse. At the same time, also produce acetyl-CoA for lipid biogenesis. So question is, how does this mitochondrial dysfunction occur? Obviously, from a ciliopathy hypothesis, we know a few mitochondria can come very close to cilia within several hundred nanometer proximity, and certainly you can have a connection through this. However, we know kidney tubular cell has hundreds or even thousands of mitochondria, depending on really the metabolic state. Seems to be this may not be adequate. Additional mechanism, I think, is probably happen in most of ciliopathy. Cilia goes through nucleus and through nuclear gene amplification, regular mitochondrial function. This is probably a very dominant mechanism for ciliopathy. But if you look at this transmission EM picture of a small region of kidney tubule, you see tons of mitochondria. For chemical regulation, you think that this mitochondria probably need to have some kind of interorganelle contact with some kind of intracellular organelle. In this case, we think it is ER. So cell biologists has looked at this connection between mitochondria and ER since the 70s. They have shown this very nice. So this is the mitochondria, as you can see, cristae. So this is the transmission EM of proximal tubule we got in our own lab. You can see ER kind of wrap around mitochondria very closely. Look at this scale mark. So this is only about 20, 30 nanometer, much closer than cilia and mitochondrial connection. And mitochondria occupy 30–40% of cytosolic volume. And depending on the studies and depending on the region of mitochondria, the surface of mitochondria could be covered anywhere between 25 to 30% by the ER membrane. And this is in a very short distance. This is called as MEMS, mitochondria-associated ER membrane. So what's the function of this MEMS? So this is a cartoon of illustration. This is ER, and this is mitochondria. So MEMS are a region of ER membrane which are kind of turned into very rigid because of the high cholesterol. In a way, MEMS is considered a little bit rough of the ER membrane. So its function is allow calcium direct transfer. We know the TCA enzyme require calcium. So one way calcium can go through is go to cytosol being picked up. The other way is direct ER mitochondrial transfer. And this is critical for TCA enzyme function. The other is really phospholipid transfer. We know most of phospholipid is produced in the ER, and there is no vesicular transport between ER to Golgi, so it has to be direct organelle contact. All right. So the phospholipid PC, PIP2, everything that mitochondrial membrane require for biogenesis all come from ER. The other very important phospholipid which is made in the mitochondria is cardiolipin. So the mitochondria cristae are expansion of inner mitochondrial membrane to increase surface area for the ETC to work. And to making this inner vegetation, you need cone-shaped phospholipid like cardiolipin. So this is what it takes to form a negative membrane curvature to form inner membrane cristae. You really need cardiolipin. Cardiolipin basically from phosphatidic acid transfer into mitochondria and to made out of it. So you can imagine without ER mitochondrial connection, mitochondria is going to be dead. So we have looked at the very detail the transmission EM of mitochondria ER relationship in the PKD knockout animal. So this is a control proximal tubule and distal tubule. As you can see, this is only small region. This is all mitochondria with cristae. And this is the proximal distal tubule. And this green arrow pointing out to ER. Again, it's kind of just wrapping with each other. So this is a precystic 4 weeks after gene deletion. You can see very clearly mitochondria number decrease. Also, the shape change, run up and become smaller in both proximal and distal tubule. Very interestingly, you see this kind of a vesicular structure. I'll come back to it if we have time. This is a very interesting structure, and this is the ER. So this is a precystic. But if we also express with TRIG-B transgene, you can see its rescue mitochondrial structure. So this is the cystic stage. You can see mitochondria is decreased, the number, and you see this funny vesicular structure, and this is ER, both proximal and distal. Distal is more fibrotic at the cystic stage, and they can all be rescued by expressing TRIG-B. So basically, we can quantify the area of mitochondria as a perimeter. We can quantify the run or flatten. So as you can see, compared to control in both area, perimeter, runness, in precystic stage, there's a reduction, and we can reverse by TRIG-B. Cystic stage or the reduction, we can reverse it for all the parameters. So you can imagine all this, the degrees in number change shape and become smaller. It's going to affect in ER mitochondrial connection. But you see some area, ER mitochondria still very in proximities. So we're kind of interested in those areas where you can see ER mitochondria cross connection names. What would be the names looks like? So that's what it looks like. So we can quantify this mitochondria, can quantify the MEMS length and the distance. So in control, this is a smaller picture, this is the enlarged picture. You can see the control, this is the MEMS coverage distance, and this is the distance of MEMS. Knockout, you have increase, and we can express trig B. We can shorten it again. So we quantify this for proximal and distal tubule. So you can see this is the distance increase, reversed by trig B, precystic and cystic. Look at the length of this length coverage decrease, and we can reverse by trig B, and cystic stage same. So we look at the proximal and distal tubule, but we know what are other tubules. Plus, we only look at the MEMS less than 30 nanometer separation. When you have a global change, it's very big, separated. There's no point to measure it. So we do need to quantify that. So we use a different assay called proximity ligation assay, where we label mitochondrial VDAC and ER-IP3 receptor. So when the two label protein come within 40 nanometer, it creates a red signal as shown here. So this is just stopping. So if you look at here, in precystic PKD2 conditional knockout, there's already marked reduction, and reversed by trig B. Same as cystic stage, there is marked reduction of a proximity ligation assay major interaction, and we can reverse by trig B. So mechanistically, this is what we think is going on. So polycystin 2 have a calcium defect, and polycystin 1, because they form a heteromonomer with calcium permeability, so they both gene mutation have a calcium defect in ER, and PGC1, the master mitochondrial regulator, transcription is calcium dependent. So this defect down-regulates the PGC1 alpha, then affect mitochondrial myogenesis. At the same time, it also affecting mitofussion, which are important for MEMS ER mitochondrial tethering. There are many pairs of a tethering protein linking mitochondria ER together, and mitofussion is one very important pair. So this is the gene data. Look at the polycystin PKD2 conditional knockout, precystic and cystic stage. As you can see, at the precystic stage, PGC1 alpha downregulated and reversed. This is control, knockout, reversed by trig B. So it's P-power alpha downstream of the PGC1 alpha, mitofussion 1, mitofussion 2, and then the proficient protein GRP1 upregulated. So this is what we think the ER calcium homeostasis defect affecting master regulating, affecting mitochondrial biogenesis, affecting MEMS structure. So I don't have time to go into this in detail, but just want to quickly show you. We also look at the same thing in polycystin 1, essentially the same picture. So PKD1 conditional knockout, we can rescue by trig B. The EM morphology or the parameters same, reduced in precystic and cystic, reversed by trig B. If we have time, we can come back if we want to look at more carefully. So basically, this is ER, MEMS, region of ER, this is mitochondria, and it's important for signal transfer, calcium, lipid, and some of the protein. So mitochondrial function is heavily dependent on the ER. And we can reverse mitochondrial function, which I didn't show, and a structure defect by expression trig B, which is the ER resident channel that regulates ER calcium homeostasis. At the same time, I want to point it out, there is another protein called sigma-1 receptor, which is an ER chaperone, pretty much restricted to the MEMS. And this is important for the formation of MEMS, also important for regulate IP3 receptor calcium transfer. And sigma-1 receptor has an chemical agonist, has been used in neurodegenerative disease in phase 3 trial. We know now a lot of neurodegenerative disease, Alzheimer's, Huntington's disease, is believed due to mitochondrial defect. And this has been in a phase 3 trial and work in animal. So we look at it in the PKD. So this is a PKD1 conditional knockout. So this is a knockout if we treat it with statin versus pre-084, which is an activator agonist for sigma-1 receptor. So you can see there is a significant improvement. So basically, we have shown this is not just improve the growth histology, also show improve in the EM image and functional. I don't have time to show you. So basically, mitochondrial connection affecting the mitochondria and health and function. Does it affect mitochondria metabolic reprogramming or not? And what is the mechanism? So before we get into that, I want to review. This is it's well known mitochondria drive epigenetic process. As you can see, mitochondria produce acetyl-CoA, which is actually the substrate for histone acetylation. Mitochondria metabolize alpha-ketoglutarate, succinate, fumarate are actually either regulator or substrate for histone demethylation or DNA demethylation. So you can imagine the mitochondria defect is going to affect in the epigenetics. So we know the decreased mitochondrial function normally should decrease acetyl-CoA production, but in PKD, because of glutamine amyloidosis actually end up increase acetyl-CoA. I want to tell you this histone acetylation affecting one of the very important genes that play in the metabolic reprogramming ADPKD. So this is a study published 6 years now by me et al. In Nature Metabolism article. They talk about CDK7 cycle-independent kinase is a transcriptional kinase important for stabilization and assembly of super enhancer for ADPKD. So the shown CDK7 gene is upregulated in ADPKD, and if you inhibit the function of CDK7, it improves the cystogenesis and metabolic reprogramming. So this is a cartoon taken from the commentary by Boletta, basically showing a lot of metabolic reprogram gene like bacteria operon, forming kind of a operon enhancer. And this is THZ1, which is CDK inhibitor, can improve cystogenesis and metabolic reprogramming ADPKD. So again, they show the PKD mice, CDK is upregulated, and you can use this inhibitor, improve cystogenesis. This is the cyst index shown in this paper. And indeed, we can confirm that in PKD1 conditional knockout, CDK7 transcript is upregulated in pre-cystic and cystic. Importantly, this is we can reverse it by expressing TRKB, again, restore calcium defect in the ER. And we can use MEMS activator, pre-084, to reverse it. So pretty much this supported our hypothesis that mitochondrial disconnection drives the CDK7 upregulation. And this is important for metabolic reprogramming in ADPKD. And how this happened? I'm going to show you this is through mitochondria-driven epigenetic rewiring because of mitochondrial disconnection. Okay, so if you look at the gene database, this is a CDK7 gene located here. And if you look at the surrounding region, the enhancer region, there are area where there is heavily histone acetylated. So we kind of identify 13 areas and make the PCR primer to probe this DNA sequence. and do a chip qPCR. So all 13 regions we probe, I just want to focus you on the C5 and C10 region. So this is a PKD1 conditional knockout, PKD conditional knockout. We look at precystic, which is the purple, red, which is cystic. So in C5, you can see control from precystic to cystic. The histone acetylation in the C5 region upregulated. And this is also occur in PKD2 conditional knockout. So is C10, as you can see. But how do we know this is really the enhancer region for CKD or CDK promoter? So this is what the study to show that. So this is if you make a PCR using the primer, one anchor to the promoter, one anchor into the distant enhancer region, and they are far away, and if you making primer on the same direction, they should not PCR, right? Too far away and the primer go in the same direction. The only way for is to PCR, you have to form a promoter enhancer loop, and you happen to form a loop and you cut it, it may be allow you to have a PCR. So this is called confirmation, chromatin confirmation assay. So we did this. So we look at C5 and C10 using C5, C1 as a control. So the chip chromatin confirmation assay, we show the control assay. So basically primer anchored into C promoter, and then the other second primer spread along the distance of the enhancer region, anywhere from the 40 kilobase to several megabase. So in the control, you can see these two regions within C5 and C10. You can PCR. That meaning this region form a loop with the promoter, and this region are enhanced in PKD1 conditional knockout. So quantitatively, C5 and C10 in precystic and cystic, this enhancer region are upregulated, and we can reverse by TRK-B and or by MEMS activator. So then we taking out the distal convoluted tubular cell line. We do a CRISPR-Cas to delete C5 and C10, and you can enhance the region. You can see CDK expression reduced. So pretty much we have provide evidence to support mitochondria.
Could you try again?
Sorry, my Siri is picking up my. So mitochondria-driven increased histone acetylation of CKD enhancer in C5, C10 region is mediated by ATP-KD by metabolic reprogramming and histogenesis. A few last slide, I want to show you the methylation. Again, we talk about acetylation. Mitochondrial dysfunction is going to change in this alpha-ketoglutarate 6 and fumarate is going to change methylation. So we did the methylation array, and out of a top 500 differential methylated profile, about 25 upregulated, increased methylation, 75 decreased hypomethylated, and we can reverse this by TRK-B. This is precystic, and this is cystic. Because we identify PKD1 and 2 conserve a mechanism, so we overlay PKD1 and 2. So out of this so many differential methylated gene, we pick out two to emphasize here. One is the PPM1K, which is mitochondria phosphatase. The other one is the ZBTB1. So this is hypomethylated, increased, hypermethylated with the gene downregulation. ZBTB1 is just opposite, it's upregulated. So before I tell you the function of these two, I want to take one slide to break up to tell you mTOR. We know mTOR is upregulated in ADPKD, particularly in cystic stage. And mTOR activation is basically sensing nutrient sufficiency. The other one is AMPK as the yin-yang sensing the nutrient deficiency. But mTOR activation also can be activated by growth factor. So this is what we know. Growth factor, PS3 kinase, and everything through TSC to activate the mTOR. But the other pathway, the other essential factor, because mTOR is anabolic amino acid sensor, so you need to sense amino acid. Among these leucine, isoleucine, these are branch-chain amino acid, plays a very important role for activation of mTOR. So basically, you have to sufficient amino acid, branch-chain amino acid plus growth factor to fully activate mTOR, and this occurs in the lysosome surface. Okay, so this allows us to target branch-chain amino acid defect. Maybe can target for mTOR. Okay, so people have shown that, and we have confirmed that branch-chain amino acid catabolism is defective in ADPKD, as we see here. Control PKD1, PKD2 conditional knockout, catabolism is impaired, branch-chain amino acid is increased. So we have done quite a lot of study. For the interest of time, I don't have time to get into that. But so basically, branch-chain amino acid go into mitochondria, it's converted into branch-chain keto acid, and branch-chain keto acid dehydrogenase is going to dehydrogenase turn into acetyl-CoA, feed into TCA cycle. And this dehydrogenase is regulated by mitochondrial phosphatase, ppm1K, inhibited by a kinase, BCKD-DK. So we found this is downregulated in ADPKD, so that decreased the dehydrogenase activity, allow branch-chain amino acid to accumulate it, and this activated mTOR, causing, along with growth factor, causing this cell proliferation. But mTOR also activated ATF4, which is very important for transcription upregulation of ASNS. ZBTB1 is upregulated because of the hypomethylated, and it turned out this is a transcription co-activator acting with ATF4 for ASNS upregulation. So basically, we think the mitochondria, mitochondrial disconnection, changing methylation, affecting ppm1K, affecting branch-chain amino acid, affecting ZBTB1, upregulate ASNS, along with CDK7 from the histone acetylation, contribute to metabolic reprogramming. So unfortunately, there is no chemical activator of ppm1K, but there is a chemical inhibitor of this kinase, which inhibits the dehydrogenase activity. I don't have time to show you. We have preliminary data. BT2, which is this inhibitor, actually increase branch-chain amino acid, or decrease amino acid, reverse the increase in branch-chain amino acid in PKD mice, and decrease mTOR and decrease cystogenesis. So basically, I just want to come back to tell you second last slide that targeting branch-chain amino acids will be a potential additional pathway to target mTOR, because rapamycin has been used, worked in animal, but in human is not effective in part due to the toxicity. So targeting branch-chain amino acid may be another additional mechanism. So summary, so we believe ER calcium defect, both polycystin 1 and 2, affecting ER mitochondrial connection, affecting the calcium transfer, causes a functional defect of TCA enzyme. Cytosolic calcium regulates PGC1 alpha, affecting mitochondrial biogenesis by affecting mitofusion to affecting MEMS. This kind of amplify the mitochondrial dysfunction and feed into epigenetics and feedback to further amplify cystogenesis. Obviously, I have shown you we can reverse this by TRIG-B and MEMS activated pre-084. We know polycystin is everywhere. We know we talk about this is important, but this is by itself is not sufficient. We know IP3 mutation is not linked to to the PKD phenotype. So the calcium defect is not sufficient. It has to be polycystin 2 function in cilia and basal lateral membrane, and together it's going to contribute to cystogenesis. So these are the people really do the work, and Susan, my colleagues, has been helping us with a lot of tubular dissection and metabolic assay in the tubule. And really the consortium and many colleagues has provided the reagent, and thank you everyone. And I got a family support here. Question?
Are there any questions for Chou? Maybe I can start. There, you have a lot of changes that occur in these pathways, but do you think calcium is central to a lot of the epigenetic changes that you're seeing? Or is it more complex than that?
Yeah. Well, I mean, the Roundtable has a paper in KI reported, CMYC and everything. There's a lot of super enhancer epigenetics, right? And she actually showed in a cell line by putting the mitochondria substance that help mitochondrial function, reverse it, And in animal, we kind of show consistent with that, but it's come from the calcium because we can reverse by TRIG-B. So we think the ER calcium defect affecting mitochondria, and then this driving some of the mitochondria-driven epigenetics reprogramming. I'm not saying this is the only pathway, but the fact that we induce TRIG-B expression at the same time we induce PKD2 or PKD1 knockout, and the reversal is 50 to 80% is not complete, and so I think it clearly plays a very early important process. But again, I don't think calcium is it's one of the very early event, probably not the only event.
Okay.
Yes.
Would you like to go ahead? You have a question?
Sorry, can you hear me?
Yes.
I still remember the paper you're referring to, which came from Professor David Clapham lab about the ion conductivity of the polycystin channel, and I believe that was a patch clamp on the cilia.
Mm-hmm.
And it was from cells, but the post-translational modifications of the polycystin was not really identified when they measured the conductance. I mean, polycystin get phosphorylated a lot, and phosphorylations, they really regulate the probability of opening, the pore size, and all of that. So this is not really trivial. And and that was only on the membrane polycystin. The cilia and the cell membrane. I don't recall that they did any patch clamp on the ER polycystin to see whether it's really more sodium or potassium than it is calcium. That on one hand. On the other hand, so the mitochondria problem basically has been there for people have been talking about it for a long time, and so you would think, I mean, there are now maybe one or two papers talking about infertility in polycystic kidney disease, because when you talk about mitochondrial problem, and you would think that you will have like a much more severe sperm motility defect because also the polycystins are present in the sperm, and also you have a lot of mitochondria there that drives the sperm motility. So do you think like the mitochondrial defect is only present in the renal tubules, or it's also present like everywhere polycystins are present, like in the bile ducts, in the pancreatic ducts, in the sperms, in whatever they are?
All right. Okay. Well, thank you for the question. Regarding the patch clamp of polycystin 2 function, yes, it's done primary in the primary cell. The native cell is predominant. The only way we can record it is from the cilia, right? And there has not been a recording of the ER polycystin function by patch clamp. But Young Yu and us has been able to express in oocyte, but that's not by patch clamp, because the expression is so low. We do a 2 electrovoltage clamp, and they do have an increased calcium permeability. But relatively, even if we look at the calcium versus sodium and potassium, clearly the ratio is not 40-1 like what Clapham has found in the cilia, but it's potassium is higher than sodium, then higher than calcium. And I think this is on cell surface membrane in oocyte by 2 electrovoltage clamp has been shown. And then the polycystin 1 and 2 heteromultiple does increase calcium permeability. So multiple groups.
And that's you're talking in xenopus oocytes.
Yeah, in xenopus oocyte. Yeah.
And that's
Unfortunately,
co-expression of this is co-expression of PC1, PC2, or only PC2?
Actually, both Young Yu and us to study both polycystin 1 and 2. And when the polycystin 1 and 2 together, they do increase calcium permeability. So these are there are two very nice papers has shown that, you know. So in terms of the effect on sperm, I think this goes to fundamental question of cystic kidney disease. We know polycystin is ubiquitous, right? But the phenotype occur in the kidney because this autosomal dominant disease, you need a second hit. It's a very infrequent stochastic event. You only need a 1,000 mutation to compress the kidney, cause the phenotype. It does occur in the liver and pancreas and other tissue, but it doesn't cause a phenotype because of the rare event. That's my understanding. I mean, some of the experts maybe can add to that.
I think the last discussion that went between Professor Harris and Germino and some other big guys in the field, I think they kind of tend to conclude that it is more the threshold of the protein levels rather than a second and a third hit. And that's where the RCRC mouse model came into the scene, and it's being like so asked for every time a grant is reviewed. But yeah, I think it's just like with aging, you have less expression. So even if you have one allele that's still functional, and over time, its expression it gets lower and lower until you go below this threshold, and then you get the manifestation. And aging, I think, should be uh, more or less. I mean, uh, well, maybe some organs they have stem cells, but other organs they don't. Like we know a kidney, they don't have stem cells, or at least till now.
So let other people ask some questions.
Yeah, sure.
So, Chou, there's a question in the chat before you get to Dr. Hollows. In your experience, which of the targets is most effective in reducing cyst volume if treatment is done in the adult stage?
So
So-
say that again.
In your experience, which of the targets is most effective in reducing cyst volume in the adult stage of the disease?
Well, I think there are many people in the audience probably know more than I do to answer that. I can tell you I mean, we have looked at the ER mitochondria, and we know that when we target pre-084 with MEMS or the ER by TrkB, we all target very early on. We haven't done like after cyst formation and try to kind of therapeutic to do it after cyst. This will be future work. But like in TrkB, we kind of a conditional transgene, activate the recombinant transgene along with when the gene is, you know. So I don't think I'm in a position to comment which is a better target, you know.
Okay, thank you. Dr. Hollows?
Yes, thank you. Very nice talk, Chou. So I was hoping you could clarify for me the relative roles of TrkB and PC2 in the ER membrane, and the data you showed is very elegant, and that TrkB expression can complement or reverse a lot of the effects of polycystin 2 and even polycystin 1 conditional knockout. And also, did you also look at re-expression of polycystin 2 in this setting to more directly complement the initial knockdown and see, do you get similar effects on mitochondrial morphology, distances, and some of the other readouts you looked at? I was just curious about the roles of these two potassium conducting channels in the ER.
All right, okay. Yeah, it's a very interesting question. We believe there are two polycystin 2 population in the ER. Obviously, I don't know anyone has quantified the relative stoichiometry, but you can imagine polycystin 2 probably way more than polycystin 1. So if you take that assumption, you probably would imagine there are two populations. One is polycystin 2 homotetramer, which the functions probably predominant conduct potassium for potassium calcium exchange to allow calcium to either transfer to mitochondria or go into the cytosol to does its work. There is another population which I think is the polycystin 1 and 2 heteromonomer, you know. So again, Yong-Yu and us both have shown that this has increased calcium permeability, but is this function identical to polycystin 2 homotetramer by facilitate calcium release from IP3 receptor or not? I don't know that, but you can imagine, again, I'm then going to speculation, in the setting of a polycystin 1 and 2 heteromonomer, and the calcium release perhaps is less than IP3 mediate calcium release facilitated by TrkB or by polycystin 2, right? Which is kind of a exchange allow IP3 to work. That's probably much higher calcium concentration versus then calcium permeated by polycystin 1 and 2 heteromonomer. But again, I'm speculating. perhaps this population of calcium might regulate polycystin 1 CTT cleavage, because locally where polycystin 1 CTT is right there, and it's going to allow the translocation that what Mike Kaplan has shown that regulate mitochondrial function. So it's in part depending on the CTT function or the polycystin 1. Again, it's speculation, but regardless, as long as calcium help, and that's why you can see the TRIG-B either way can help. And doesn't mean polycystin 1 and 2 are identical. I know the current thinking is other than the timing, the phenotype seems to be identical, but we know they are not identical. The expression pattern, temporary, and then the subtissue. So whether the growth phenotype looks the same, does that mean they work identical or not? I mean, you can tell me. I mean, many experts here, but we can see reverse of polycystin 1 and 2 by TRIG-B. Doesn't mean they work identical, but the calcium is probably all part of a story. So I guess to answer your first question, your second question, also yeah.
I was just wondering if you tried re-expressing polycystin 2 to see if that also rescues, like TRIG-B.
No, we haven't done that. I mean, the Somlo and then Patel, they do a lot of re-expression of polycystin. We haven't done that.
Thank you.
Hey, Jean, we have a couple of minutes left. You have a quick question?
Yeah, I was just wondering whether you think PC1, PC2, or PKD1, PKD2 mutations directly break the MEM structures. And what is, I mean, your work is very interesting, and and indeed the phenotype seems to occur very early, and was pre-cystic, right, before the kidney has structural changes. And did you mention already, like, what's the connection with cilia, or it's independent pathway, or it's downstream, or it's what's the relationship?
So based on the data that we can reverse by TRIG-B, we think a part of the function is definitely through calcium signaling, and we know it's also very well known PGC1 alpha and mitofusion is all calcium dependent. And so it makes sense to think in the calcium homeostasis defect affecting the PGC1 alpha, affecting mitogenesis, and that's why you lose the ER mitochondrial connection. And through mitofusion, you affecting the MEMS tethering, right? So that's why we think. Can we say polycystin does not have additional function that directly affecting MEMS? We don't know that, and we only based on what we can reverse it, So the other possibility, can calcium directly, instead of going through CAM2 kinase, calcineurin, and PGC1 alpha, can have a calcium have a direct affecting the interorganelle formation. It's possibility consistent with our data. We cannot exclude that. So your question, does polycystin has a direct effect on MEMS? Certainly could be. I cannot comment on that. Yeah.
Thank you.
Okay, we're right at the hour. Chou, I want to thank you for a wonderful presentation. It's very interesting. And I will see everyone on May 4th for the next roundtable. Bye, everyone.
Thank you. Yeah.
Thank you.